Hello, learners. Welcome to the License lecture series. I'm here to simplify biology, but also keeping the science intact. So today we'll be exploring how bacteria quickly converts its genetic information into action through the process of prokaryotic transcription. So keep going and let's explore. Bacteria, they live in a constantly changing environments. To survive, they should respond faster and fast like our cells, much more faster than our cells. That rapid response begins with a highly efficient process called as transcription. So transcription, what is transcription and why is it needed in prokaryotes? Transcription is the process of copying information from DNA into RNA. RNA it acts as the working message which directs protein production. For prokaryotes, this process it should be energy efficient and extremely fast because their survival depends on the rapid growth and quick adaptation. So where does transcription occur in bacteria? Bacteria they do not have a nucleus. So their DNA lies freely in the cytoplasm. As a result, transcription also occurs in the cytoplasm, allowing RNA production and protein synthesis to happen simultaneously almost at the same time. Now let's see who is the hero of this story. The hero is the RNA polymerase, the central enzyme for the transcription. This enzyme reads the RNA template and synthesizes so this enzyme reads the DNA template and synthesizes an RNA molecule. So in bacteria, a single RNA polymerase carries out all the entire transcription, and it makes it a very powerful and efficient molecular machine. So let's see who is the. But however, RNA polymerase cannot just identify genes on its own. So it needs a helper, it needs a sidekick. So that's where sigma factor comes into place. Sigma factor helps the RNA polymerase, to recognize the correct start site of a gene. Without sigma factor, transcription would become inaccurate and inefficient. So sigma 70 is one of the most common ones. Now let's see how they work together, right? So when the polymerase binds to the sigma factor, they form what is known as a holoenzyme. A holoenzyme is a complete enzyme which has your protein part as well as another enzyme, the coenzyme with all subunits, and then there is a protein subunit which help in its activity or in its process. So this particular holoenzyme, it searches for the DNA promoter sequences. So once the transcription starts, the sigma factor will detach from it and the transcription begins. Now let's see how that happens, right? So the holoenzyme will go and bind to a specific DNA region, which is known as promoter. A promoter is really important because it's a specific DNA sequence which signals the starting point for transcription. The so the in bacteria, the promoters have a conserved region, which is -10 and -35 regions. So it's at these regions, or these sequences, are the molecular signals which help guide the RNA polymerase to the correct transcription start site and initiate transcription. So, in prokaryotic transcription occurs in three major stages. Initiation, where the RNA polymerase binds and starts the RNA synthesis, and the second, which is elongation, where the RNA strand is extended. Third is third is termination, where the RNA synthesis is it is complete and the transcript is released. So this is the three main process and we'll be exploring each in detail. So let's begin. Okay, now initiation. During initiation, the holoenzyme binds to the promoter and the DNA double helix. It starts to unwind near the -10 region. The first RNA nucleotides, so sequences are joined together and the transcription officially starts. Okay. Now let's move on to the second stage. So this is the series of steps that happens in or occurs in transcription. Sorry, in the initiation step. So you have the holoenzyme formation and promoter binding. Then there is the closed complex formation, and finally there is this open complex and the about to initiation, which is where a short stretch of RNA sequence made. Okay, and then yeah. Now let's begin. Now elongation, after initiation, the sigma factor dissociates. The RNA polymerase moves along the DNA template strand and it starts to add RNA nucleotides one at a time in the 5' to 3' direction. So the RNA strand grows as the enzyme progresses forward. And in elongation, so RNA polymerase is really fast. It works rapidly. It starts advancing at around 40 to 50 nucleotides per second. That's really fast. And also it is efficient. It also performs on proofreading function. So what two kinds of proofreading it does is pyrophosphorylatic editing, and the second is hydrolytic editing. So this is also crucial. Okay, while the synthesis happens, it also makes sure that it doesn't incorporate any wrong base. Okay, so that this is also critical. Now let's move on to the end part. So transcription should be stopped. It should stop at a correct point. Okay, and that point or that sequence is called this process. This is what termination is. Okay, so you have sequences known as terminators which help dissociate their DNA and release RNA chain which it has made. So because bacteria, it has two termination processes or two termination mechanisms. The first is Rho-independent and the second is Rho-dependent termination. So the Rho-dependent termination is where there is a protein called as Rho factor which helps to induce termination, and the second is Rho-independent termination, where, as the name suggests, it doesn't use any kind of protein, but instead there is a structure that it forms like a hairpin which causes the termination. Now let's explore each little bit. So the Rho-dependent termination, here there's a protein called as Rho which helps to bind to the RNA transcript. It's like an accessory protein. So the Rho factor moves along the RNA and it catches up with your DNA polymerase. It then causes the polymerase to release the molecules. That is the whole process here. Fine. And now moving on to the Rho-independent termination. In here, the RNA forms GC-rich hairpin loop, which is followed by a series of uracils. This structure destabilizes the RNA and DNA complex. RNA polymerase releases the RNA and it detaches from the DNA. So what happens in the end result? So the final product that you have out from all these three processes is that you have a functional mRNA. Okay, and this functional mRNA helps, so it goes into the so ribosomes come and start translating this mRNA. Even before the transcription is fully complete, the protein synthesis has also started. Okay, because as I told you, there is no nucleus, right? So the both processes mechanisms happen simultaneously. Okay, so yeah. Now, so prokaryotic transcription, finally, it is a master class when it comes to speed and precision. Why is it that? Because it doesn't use, there's no nucleus. It's an energy efficient process. It uses only essential components. So that, okay, and that is good. And precision because it uses promoters and sigma factors. They'll make sure that the gene transcription or the gene expression happens accurately. Together, these features make prokaryotic transcription a master class when it comes to biology efficiency. Now, what are the applications when it comes to prokaryotic transcription? So, understanding bacterial transcription has major practical applications as well. First is bacterial RNA polymerase is a key antibiotic target with drugs like rifamycin, which helps to block transcription in harmful bacteria. Second is engineered bacterial promoters and sigma factors. They allow scientists to precisely control gene expression in biotechnology. And the third is that bacteriophage RNA and the bacteriophage RNA polymerases, T7 RNA polymerases. These are all powerful tools which is there for high level gene expression and RNA production. So these are the images. All right. Now let's do a quick recap and a memory boost, right? So transcription is a process where it copies the DNA into RNA. Okay, that's the first step. Second step is RNA polymerase is the core enzyme. And then there is a sigma factor which helps to guide the promoter recognition. Transcription occurs through initiation, elongation, and termination. The system is fast, efficient, and highly precise. So with this, we have uncovered how bacteria efficiently express their genes. In the next video, we will be exploring how transcription becomes more complex in eukaryotic cells. Until then, this is Life Science Lecture Series, Learning Life one concept at a time. Thank you. Signing off.